Good afternoon, students. On the previous day, we were talking about the determination of the bacterial concentration in any given water sample. Among various methods available, most probable number is another way of expressing the determination of the bacterial concentration in the drinking water or any other water sample. So first of all, let us understand what is the meaning of this most probable number. As the name itself indicates, most probable number means the number is going to repeat frequently when the sample of water is subjected for detailed analysis. So it goes like this. In order to arrive at the number of coliforms in a water sample, it is necessary to notice the positive results from various size portions of a sample. So here I would like to explain what is these various size portions. Normally the procedure goes here like, see, there will be a set of 5 test tubes in which the procedure says that in each of these, say we are pouring 10 ml of water sample in each of the total 5 test tubes, so that the total quantity of water sample poured into this first set of 5 test tubes will be 5 into 10 ml each, so 50 ml. Similarly, for another set of 5 test tubes wherein we are pouring only 1 ml of the water sample to each of these 5 test tubes, so that the quantity of water sample taken together for this second set of 5 test tubes will be 5 into 1 ml, that is equal to 5 ml. In the same way, in third set of 5 test tubes, only 0.1 ml of water sample is dispensed, so that the total quantity of the water sample dispensed into the third set of 5, it will be 5 into 0.1 ml, it is 0.5 ml only. And the total quantity of the water sample dispensed for these 3 sets will become what? In the first set, 5 into 10 ml, 50 ml. In the second set of 5 test tubes, it will be 5 into 1 ml each, therefore 5 ml. And in the third set of 5 test tubes, in which 0.1 ml of sample was poured, it will become 5 into 0.1 ml, therefore it is 0.5 ml. If you add all these, the total quantity of the water sample dispensed for all these 3 sets will become 50 plus 5 plus 0.5, 55.5 ml. So here, when this water sample has been poured into these different sets of the test tubes, in which already there will be some nutrient medium was present, over that nutrient medium we are dispensing these water samples, and after pouring these water samples over this nutrient medium already present in the test tubes, it is now the mouth of all these test tubes is covered with the sterilized cotton, and these sets of 5 test tubes are kept in a temperature controlled unit that is called incubator at, say, some designated temperature set, and it is kept there for the predetermined time, say it may be 24 hours or 48 hours at temperature maybe 35 degrees or 37 degrees as designated, prescribed in the procedure for conducting the test. So if at all any bacteria are present in this sample of the water taken, then by the end of this incubation period, say 24 hours or 48 hours, there could have been a fermentation process taking place within the test tubes. These bacteria taking the nutrient medium as their food source, they multiply in their number, and in this fermentation process, the result is observed that the gas formation. If there are any bacteria, then only they can ferment this food and gas is going to be formed. If in any set of these 5 test tubes, if there is a gas formation, then we can call, so the test is positive. And say for example, in the first set of test tubes where 10 ml sample was poured, say 2 samples they indicated the gas formation, then 2 out of 5 are indicating positive, obviously remaining 3 are negative. Say in the second set of 5 test tubes where 1 ml quantity of sample was taken, if 3 test tubes are showing the gas formation, then 3 are positive, then 2 obviously will be negative. Similarly, in the third set of 5 test tubes, if only 1 test tube is showing the gas formation, then 1 is positive, obviously remaining 4 test tubes they indicated negative. So we will have to calculate the number of positive portions, and then we will have to also calculate the number of, the ml in all the negative test tubes. Means ml means it is the quantity of the water sample taken where the test result was found negative. In the first test, for example, 2 are positive, obviously 3 are negative. So therefore the quantity of the sample taken in the negative test tubes is equal to 3 into 10, that is 30 ml. So like that we need to add in the remaining sets also. Okay. So the meaning of this most probable number is that, so the most probable number or MPN of coliforms in the water sample is obtained by applying the laws of the statistics to the results of the test. The MPN of coliform or E. coli is defined as that bacterial density, if it actually present in the water sample which is subjected for examination, would frequently given the observed analytical result. Means if when the water sample is examined for this bacterial content or concentration test, the result obtained will be, that value will repeat several times, therefore it is called as most probable number. In other words, MPN indicates the bacterial density which is most likely to be present in the water. The standard sample for a potable water is 5 10 ml portions. In all these, if all these are negative, then MPN is said to be 0, because in the equation there is a term number of positive portions, where number of positive portions become 0, entire result would become 0. I will show the equation now. If all the samples are indicating negative, then the MPN value will become 0. Suppose if only 1 out of 5 sample is positive, then MPN is say 2 per 100 ml. I am going to demonstrate this in the next example. So one water scientist by name Mr. Thomas, he has suggested the following approximate equation for the calculation of the MPN. So as per him, MPN is equal to 100 into number of positive portions in the numerator. In the denominator, ml in all negative portions and ml of all portions. Did you understand? So MPN, most probable number, is equal to 100 multiplied by number of positive portions in the numerator divided by within the square root ml in all negative portions, means the quantity of the sample in the test tubes which showed negative. We need to add all portions like 10 ml or 1 ml or 0.1 ml, we added accordingly. That quantity should be multiplied by ml of all portions again in the denominator. ml of all portions means, for example, if if 1 out of 5 10 ml portions is positive and 1 out of 5 1 ml portions is positive, then MPN will become obviously the number of test tubes indicating positive results are the 2. Therefore, in the numerator we have, as per the formula, 100 into within bracket 1 plus 1, where first 1 indicates it is 1 out of 5 test tubes where 10 ml was poured. It indicated one of these 5 indicated positive. Similarly, in the other set where 1 ml each in the 5 test tubes sample was poured, there also 1 test tube indicated positive. Therefore, number of test tubes indicated positive values will become now 2. And in the denominator, say ml in all negative portions. In all negative portions means we may use a combination of different proportions or portions, like say 10 ml and 1 ml, 0.1 ml, 0.01 ml, like this, depending upon the test and its importance. So ml in all negative portions means when one test tube containing 10 ml sample was found positive, obviously the remaining 4 test tubes where 10 ml sample was taken, they will become negative. So 4 into 10 ml in each of these failed or negative test tubes, therefore 4 into 10 it is equal to 40 ml. And also in the second set where only 1 ml water sample was poured for each of these 5 test tubes, so 1 test tube indicated positive, means gas formed it was noticed only in 1 test tube. Obviously the remaining 4 test tubes indicated negative. Therefore the quantity of the water sample which showed negative, it will become 4 into 1 ml, therefore it is 4 ml. So ml in all negative portions means 4 negative where 10 ml was poured and 4 negative where 1 ml was poured. So therefore 4 into 10, 40 ml plus 4 into 1, so it is 4 ml, total it is 44 ml. And the next term in the denominator is ml of all portions. Ml of all portions means it is the total quantity of the water sample poured into different sets of the test tubes. Therefore in the first set 5 number of tubes were 10 ml each, therefore 50 ml, and second set of 5 test tubes where 1 ml was dispensed, therefore it is 5 ml. So ml of all portions indicate 55 ml. So if you simplify after substituting in the given formula, the result is going to be 4. It means that the result is 4 coliform bacteria per 100 ml of the water sample. So this is the result and the way of expressing the number of coliform bacteria present in every 100 ml of the water sample. To declare the test as negative and to certify that the water is fit for drinking purpose, the MPN value should be 0. It should be 0. If it is more than 0, that is any positive value indicates there are the presence of the bacteria and some of them may be pathogens too. So we will have to detect for the presence of the type of bacteria. If such kind of bacteria are not harmful, then you can, just for the safety purpose, you can disinfect the water sample. Generally at the end of the treatment plant, that filtered or the total treated water is disinfected using different methods and then it is supplied to the consumer or the public.